Good morning, everyone. Actually, it's my honor to join such a community of researchers and families trying to understand white Sutton syndrome. My name is Azza Soliman, and last year PhD student at Institute of Human Genetics in Mainz in Germany. And actually, for the last 5 years, I was studying POGZ mutations and white Sutton syndrome. So today I'm going to share with you our recent findings, and I hope this could help in understanding more white Sutton syndromes and how POGZ mutation can affect the brain development. So as you might know, this is the human POGZ protein with the different domains. And this picture is actually from a publication from 2020, which shows the different mutations that have been identified in patients with white Sutton syndrome. And you can see that the mutations actually spread along the protein and the different domains. And down here you can see patients with white Sutton syndrome. And also, as you might know, that there's no specific clinical phenotype of white Sutton syndrome, but we can also say that they have still a bit of specific features like facial features and also the effect on the brain development and sometimes even the movement development. So in our lab, we mainly follow using the IPCs or induced pluripotent stem cells, and we start with a healthy donor where we could isolate fibroblasts, so specific skin cell type, and we reprogram those fibroblasts into IPCs, induced pluripotent stem cells. And what is specific about those cells that they can actually become any type of cells. So with the help of CRISPR-Cas technology, which is a genome editing tool, I could induce a mutation in POGZ and then having what we called altered IPCs. So IPCs that have a mutation in POGZ that it induced with CRISPR-Cas. On the other hand, we could also have a patient who already has white Sutton syndrome and has a mutation in POGZ, and then we also isolate fibroblasts and then reprogram those fibroblasts into patient IBCs in that case. And then either with the patient IBCs or the altered IBCs, we induce or differentiate them into neuronal cells, which in our case, neural progenitor cells, NPCs. And then we could either go into deculture to answer some questions or into more 3D models, so mimics more the physiological environment, which in my case, I use neurospheres and cerebral organoids. So having this protocol, I ended up having two different IPC cell lines. One is altered. This is the help of CRISPR-Cas, and in that case, it was a healthy female donor. So the cells are female cells, and it has a mutation in exon 2 of POGZ, and it was one nucleotide deletion that leads to frameshift mutation and a truncation of the protein 8 amino acid number 29. This is actually here. I put it here. This is like, as you can see, it's really close to the N-terminus of the POGZ protein. The other cell line, it was a patient IBCs, and it's a boy, so it's a male cell line that has a mutation in exon 17 of POGZ, and it was one nucleotide duplication and also frameshift that will lead to frameshift mutation and truncation of the protein and amino acid number 40. And here you can see this is other red mutation, and you can see that it's located actually in an HPZ domain. So this is a domain that binds to the HP1 alpha. So the first thing that we wanted to investigate actually is the effect of these two mutations, so those two frameshift mutations in the N-terminus and in the HBZ domain on the protein level and the nuclear localizations. So the first thing that both of the mutation actually affect the protein expression. So you can see in the female that the mutant MBC showed almost half of the protein expression, and also in the patient MBCs compared to the wild type. For the localization, it's known that POGZ localized in the nucleus, or mainly localized in the nucleus. And one thing that we wanted to investigate if this mutation might affect the protein localization. And as you can see that in the mutant female, the CRISPR-1 MPCs, the localization didn't change. It's still co-localized where the DAPI is, and it was the same case with the patient male MPCs that POGZ still localized in the nucleus, especially that we still have the wild type POGZ expressed from the wild type allele. The next thing that we wanted to investigate if this mutations actually could affect the differentiation. So the MPCs are neuroprogenital cells that can actually or can or should differentiate to mature neurons. And that's what we tried to investigate in 2D culture. So we have the MPCs, we induce them to differentiate into neurons by changing the media for 2 days. And then after 2 days, we fix the cells and stain for MAP2. And MAP2 is a marker for the mature neurons. And then count how many MAP2 positive cells are present. So in the case of the female MPCs, we could see that there was a significantly higher number of MAP2 positive population compared to the wild type. But this effect was not there when we compared the patient MPCs, the male patient MPCs, compared to a wild type, which indicate that actually the neurodifferentiation is enhanced, but only with the POGZ mutation that it's located in the N-terminus in the female cell line. Next, we wanted to study this more into 3D models. So in that case, I used neurospheres. And what we wanted to see first is the effect of this mutation on the proliferation. So, and in case of the neurosphere, that did in terms of the diameter of the neurosphere. So I would culture the neurospheres for 5 days, and then after 5 days, I take pictures and then measure the diameter of the neurospheres, and then the diameter. This can be indication for the proliferation and how much the cells can proliferate or self-renewal activity. And as you can see in both cases, in the female and male mutant neurosphere, that there was a significantly decrease in the diameter of the neurospheres compared to the wild type. So the corresponding wild type, they were smaller neurospheres in the mutant neurosphere were smaller compared to the wild type. What we did next, it's also trying to study the migration, which is a really important process during the maturation and differentiation of the neurons, that they start to migrate to the upper layer. But we wanted to investigate actually this in 2D culture. So in that case, we plated the neurospheres in a plate, so an adherent environment, and we induce the neuron differentiation. So we push the MPCs of the neurospheres to become neurons by changing the media, and then for 2 days, and then the cells started to migrate outside of the neurospheres, and then measure how far this cells would migrate from the surrounding or the area or the surrounding of the neurospheres. And as you can see that in both cases, also in the female mutant and the male mutant neurosphere, that the cells or the neurons started to migrate at a significantly longer distance compared to the neurons migrating from the wild type neurospheres. So what we could see from this, that POGZ mutations both in the N-terminus and in the HBZ domain actually impairs the self-renewal activity, but on the other hand, it enhances the neural migration of the MPCs. Then we wanted to study how this mutations in the N-terminus and an HBZ domain could actually change the transcriptome of the MPCs. So we did RNA-seq. or RNA sequencing. And as you can see, of course, there was some DEGs, so differentially expressed genes, common between the mutant and mutant female and mutant male. And when we look to the GO terms, we could see that a lot of the DEGs are enriched for chromosome segregation, mitotic sister chromatide segregations in the mutant female. And this is also the case of the mutant male, related to the chromatoid segregation, but what also pop up more that it's also GO terms that are involved in the double strand break repair, and in the synapse assembly. One thing that it was interesting to see was that the transcriptome of the cells that are carrying mutations in POGZ, so the mutant female and mutant male, they had less differentially expressed genes together compared to the wild type cell lines, which means somehow POGZ mutations affect the transcriptome to the point that they are more similar, which in our case we called it a POGZ specific transcriptome. We also looked to this DEGs in the mutant female and male, and we could see that many of them actually intersect with the genes. So the autism-risk genes that are found in Safari database, which is the database that includes all or most of the high-risk genes for autism spectrum disorder. Then we wanted actually to investigate the differentiation status of the mutant NBCs using the RNA-seq data. And for this, we used a publication, so a study where they tried to investigate or intersect the transcriptomic signature of the neurons during differentiation and maturation starting from IBCs. And from this study, they come up with a list that has kind of 19 MBC predicting genes and 25 neuron predicting genes. And we look to the expression of those genes in our data or RNA-seq data. And first thing that we notice, of course, that the samples are separate according to the genotype. And if you can see here, the neuron predicting genes, here the MBCs predicting genes. And one thing, for example, that the wild type female, that they express the neuron predicting genes less than the average of the other samples. And when we did principal component analysis, you could see that clearly that the mutant cells, either male or female, they clearly separated together across the PC1, so the first principal component. And when we look to the loading genes of this principal component 1, so PC1, we could see that the higher PC1 score, it actually indicates a higher expression of the neuron predicting genes, and it was the other way around for the MBC predicting genes. So the higher PC1, that means the less MBC predicting genes are expressed in our samples. Then next we wanted to see that how POGZ mutations would affect the binding of POGZ to different genes, because it's known so far that POGZ can act as transcriptional regulator. So the first thing that we could see from our cut and tag, we did cut and tag data, that actually majority of the peaks actually intersect with the promoter regions, which again support the claim or what has been shown already, that POGZ can act as a transcription regulator. We wanted also to validate our data, so we used a publication where they used cut and run actually, and then we validate our data, and we could see that our peaks or our POGZ bound genes actually intersect with other POGZ bound genes that found in this publication in using human embryonic samples in both in the wild type cell lines and in the mutant cell lines. When we look to the GO terms for the POGZ bound genes, we started with the wild type cells, and you could see that many terms are actually common between the female and male cells, and mainly it's about axonogenesis and Wnt signaling, a regulation of the neuronal projection development, and many other DO terms that are related to, I would say, neuron development or the brain development. We also looked to if those POGZ-bound genes or bees that we found intersect with the safari genes, and we could see that the POGZ-bound genes actually are enriched for the autism risk genes, and even if it's significantly higher, observe it compared to what expected to be seen in female cell line and in the male cell line. When we look to the differential occupancy analysis, so you could see that in the female cell lines, in the mutant, that almost 700 or 6,000, sorry, 6,600, 8 peaks loss binding, and around 500 loci gain binding, and this shows how the mutation in the N-terminus affect the binding or the ability of POXY to bind the DNA and different genes. That was the case also in the male cell lines, but with way less number of the peaks. So 9 peaks showed to lose binding, and only 35 loci gain of the binding. And this actually might be explained because we had to exclude some replicates from this experiment for the male. So this might explain the fewer number of the beaks that we could actually recall. Lastly, that we also tried to see how to compare between the RNA-seq data and the cut and tag data. So, and this might answer the question that actually POGZ can directly interact with those DEGs that we saw in the RNA-seq data. So we could see, for example, in the female sample that many of the POGZ peaks intersect already with the downregulated genes, DEGs, and the upregulated DEGs. And here also you can see how it's distributed close to the transcription start site in the upregulated and the downregulated genes. And that was also the case with the male samples, where a bit less, but still POGZ peaks intersect with the downregulated and the upregulated DEGs. At the end, I wanted to go a bit more into sophisticated 3D models, although I didn't have the time. That's why I did the organoids mainly for the female mutant cells. And what you see here, this is what we call brain organoids. This is a days old cerebral organoids. And we followed Lancaster protocol. And what you can see here of the staining at SOX2 positive, where you see the pink color, and this is a marker for the MBCs, so the neuroprogenitor cells, and then MAP2 in green, as I said, which is a marker for the mature neurons. And what you see here, this what is supposed to be a circle, this is a ventricular-like structure where the MBC, so the pink cells, should differentiate and maturate to become a mature neuron, and then migrate from inside of the ventricular-like structure to the outside, the surrounding, and become MAP2 positive cells. This is a wild-type female organoid, and this is the CRISPR or the mutant female organoid. We investigated, or we tried to study of the organoids in terms of the number of the ventricles formed, so how many ventricles are formed per one organoid or the whole area of the organoid, and we couldn't see any difference between the mutant and the wild type organoid. But when we look to the area, so how big this area of the SOX2 positive or how big the area of this ventricular, as we could see a significantly higher area of the ventricles in the wild type organoids compared to the CRISPR mutant organoids. We also look to MAP2 positive area compared to the total area of the neuronal area, which is like the SOX2 and MAP2, and we could see it was almost significantly higher in the mutant organoid compared to the wild type organoid, which is again fits what we saw in the 2D culture, that the female mutant showed a significantly higher MAP2 positive cells. We did what we call dissociation of the organoids. We use a dissociation kit, and in that case, we dissociate the whole organoids into a suspension of single cells, and then we plate those cells for 2 days, and then we just stain for MAP2, which is again MAP2 is a marker for the mature neuron, and we count how many MAP2 positive cells are there. And you could see again that the mutant organoid showed a significantly higher number of MAP2 positive cells in the dissociated cells compared to the wild type organoids, which is again tells that the mutant female organoids show more what we call a differentiation or MAP2 positive cells compared to the wild type. Then we come up with a bit of hypothesis that why the phenotype we see more this MAP2 positive, this what we call more differentiation in the female cell lines and not in the male. And then we saw that because of the mutation, actually, if you remember, in exon 17, so it's a bit late in the close to the C terminal of the gene, that we might have a truncated protein. We couldn't see in Western blot that I did in the beginning, so we tried many antibodies till we could see actually a truncated protein in the patient male. Here, as you can see, that does not present in the male wild type. And this needs more investigation, but now we have the hypothesis that this extra truncated protein might be the reason why we don't see or we see mild phenotype in terms of the differentiation in the male mutant cells compared to the female mutant cells. This will bring me to the summary of my talk. I hope it was not too much, but that actually the heterozygous mutation in both N-terminus and NHPZ domain decrease the protein expression, but it does not affect the nuclear localization, and that the neuronal differentiation was enhanced in the MBCs carrying a mutation in N-terminus that was what we could see in the 2D culture. And we saw the same using 3D neurosphere models, that the proliferation was affected in terms of the self-renewal activity of the neurospheres by measuring the diameters, and that the migration was enhanced in the mutant neurospheres compared to the wild type. From the RNA-seq data, we could see that the GO terms got affected in the mutant MPCs compared to the wild type, and it was enriched for chromatoid segregation and DNA repair. And somehow that the cells that are carrying POGZ mutation, the transcriptome of them were more similar, which we call a POGZ-specific transcriptome, and that the differentiation status of the mutant MPCs were primed for more differentiation status, if I would call, and that's what we called accelerated differentiation. For the cut and tag study, we could support or prove what already has been found in other studies, that POGZ can act as transcription regulator, because most of the peaks or many of the peaks were intersect with promoter regions, and that many of POGZ peaks actually enriched in the DEDs compared to the non-DEDs, which mean that POGZ has a direct interaction with those genes. And last, that POGZ peaks intersect with post upregulated and then downregulated. And again, the support what has been found already that POGZ actually can act as post activator and suppressor for the transcription. So at the end, I want to thank you for your listening, and I want to thank you all the members of my lab in the human genetics, especially Jennifer Winter. She's my direct supervisor, and I want to also thank you, Restu. He's my support in everything related to bioinformatics and the whole lab, and you and everything. And I'm open for any questions. Thank you.